Okay. I'll go ahead and
It says I'm still in practice session. I'm not sure. But
okay.
maybe not.
I'm gonna hit start webinar.
Okay.
All right. Start.
It looks like we're good. So I'm going to go ahead and start. Hi, everybody. It's Dr. Wohler here from Integrated Medicine Academy. I want to thank Great Plains Laboratory again for sponsoring another monthly webinar. What I thought I would do in this particular talk is talk about vitamin D, but take a different angle on it. And that a lot of what gets discussed with regards to vitamin D is its support in the immune system, bone support, things like that. And I'll mention those things, but we're going to take a little bit of a different direction as we get into this a little bit more. So for those of you who have not heard me before, I've been an integrative and functional medicine physician for many years, going back to the late 90s. I've been doing a lot of clinical education for Great Plains Lab over the past many years as well. In fact, I teach their entire organic acid test seminar, but I've been doing these webinars as well for many years. So I've spoken for them in person as well as online. I've done a lot of other education throughout my career. I'm a practicing clinician. I'm also co-founder of, let me go back, sorry, of Integrated Medicine Academy, which is an online academy we have with regards to various mastery courses in integrated medicine. Medical director of Functional Medicine Clinical Rounds, which is a membership website for healthcare practitioners who are needing assistance in lab interpretation, case presentation, troubleshooting, etc. And then in my own practice, I work with individuals with autism, other special needs, autoimmune diseases, gastrointestinal, and neurological problems. So vitamin D. If you don't know, Great Plains actually has a very convenient blood spot test, a finger prick test for vitamin D. So it makes it very convenient to use, to have people have some of these test kits at home that they can test their vitamin D level frequently as needed. Because the only way to really know what your vitamin D levels are is you have to test. Okay, so very convenient in that regards. So just as an overview, vitamin D we know is necessary for many different metabolic functions. It supports the immune system, muscle metabolism, bone formation, growth and repair, helps to reduce inflammation. It regulates blood calcium levels. It's also involved in phosphorus regulation as well. And then when we get down into the cell level or the gene level, essentially, it regulates cellular proliferation and differentiation and something called apoptosis, which is important because we'll talk about apoptosis towards the end of the lecture, which is very much in the mitochondria regulated by the vitamin D receptor. Now, 25-hydroxy vitamin D, which is the combined measurement of D2 and D3, I'll define those here shortly, is the major circulating form and really a good indicator overall from a testing standpoint that gives insight into vitamin D status. So a lot of people have vitamin D deficiencies. We know that vitamin D deficiencies are known risk factors for many common diseases, from certain cancers, osteoporosis, cardiovascular disease. They figure about 70% of American kids have some either level of a deficiency or just an insufficiency or suboptimal levels. Now, low vitamin D is associated with increased risk of all-cause mortality. So that's all deaths in a population. And it's estimated that approximately 1 billion people globally exist in a vitamin D deficient state. And think about that, that's huge, huge amount of numbers of people around the world. That actually comes from an interesting article I'll point out here shortly on vitamin D and what's called the non-genomic actions of vitamin D. So the range for adequate vitamin D, you know, anywhere between 40 nanograms per ml to 80 nanograms per ml, getting above 50, some some websites or some practitioners you listen to advocate for levels above 60 nanograms per ml is really kind of considered the ideal amongst many different types of integrative medicine resources. And one of those would be the Vitamin D Council. So the vitamindcouncil.org, good website. They've got research, they have their own blog. They recommend their own sort of range, a target range essentially for vitamin D. You can always go to their website for updated information. And so a lot of times people will say, well, how much vitamin D do I use? What's the dose? Well, the problem is that there's not an established dose, right, for every single person because it's variable on various factors. Now, from a conservative standpoint, we could probably use maybe between 1,000 to 2,000, an average 1,500 IUs per 25 pounds body weight per day. And that would probably get us close for most people. You can always get a little bit more aggressive as well as needed. And so as an example range, 5,000 to 10,000 IUs daily for adults will tend to work out pretty well. But again, right, we've got to test to know for absolute certainty. And then in children, between 1,000 to 3,000 IUs daily is a typical range. But the reality, again, is that we need to check vitamin D levels consistently in order to get accurate measurements. So otherwise, it's just a guess. Now, you can do it more frequently than 3 months, or you could do every 3 to 6 months, or maybe start off every month for a period of time to get people on track and then spread it out from there. So there's a lot of ways of doing it. But one thing to realize is that in order to know for sure, we've got to test the levels. Now, there are different types of vitamin D. So 25-hydroxy D, which is the major circulating form, is our ergocalciferol, which is our vitamin D2. That's what we get from our diet, as well as supplements. And then there's the cholecalciferol, which is what gets produced in our body from sun exposure. And so the D2 and the D3 are the precursors to what's called the 125-hydroxy vitamin D. And that is the active form of vitamin D that interacts with the vitamin D receptor at the cellular level, but it has a very short half-life. So what is measured is the 25-hydroxy D because it is the best assessment overall for vitamin D status, including D that is obtained from the diet, through supplements, or is produced from UVB light exposure. So if we look at a typical vitamin D blood spot test from Great Plains, the test actually provides a lot of information. So again, it's looking at total D and then the breakdown component of D2 and D3, knowing what you get either from sunlight exposure as well as diet. And then it's got a nice reference intervals that give us an indication of anything from the optimal levels in a healthy population, which would be that 40 to 80 nanograms per ml. Those would be units we use here in the US, and then nanomoles per liter is what's used internationally. And then gives us some indication of either severe deficiency, moderate deficiency, which would be also an insufficiency or suboptimal levels, and then elevated levels, so potential toxicity. And if you read the test, it actually gives you an idea of what somebody might be risk for if they exist in these particular reference ranges for a period of time. So a deficiency state is an increased risk for osteoporosis. A severe deficiency is an increased risk for rickets, for example, which is a bone deformation disease in children. But as we mentioned before, vitamin D, we know, plays a lot of roles in health and also in deficiency or insufficiency states, various diseases, anything from cancers to diabetes to multiple sclerosis to cardiovascular problems, rheumatological issues, mental health problems, etc. So again, it's that range we're looking for, 40 to 80. My target usually is above 50. There's some docs I've listened to that advocate for 60, but if we're around that 50 range, we're doing a pretty good job, or our patients doing a pretty good job at maintaining their vitamin D levels. Now, from time to time, you'll actually get some blood spot tests that come back that are elevated. This actually was a case from my practice, which was actually in a child where they were using high dose vitamin D for a period of time, and the levels came back high. There wasn't any detectable problem that was occurring because of that, but we did make adjustments in the amounts that were being used. And again, this was another reason why we need to test people frequently, because they may not recognize that they're taking too much. And nowadays with COVID, right, there's a lot of talk of different supplements, taking zinc, taking vitamin D, et cetera. So there's a lot of recommendations out there. And so sometimes people can be taking excess amounts of these things for prolonged periods of time and just not know it. So we can do them a favor by making sure that they're testing their D levels through this convenient finger prick test right at home. Now, some of the main issues that are actually related to vitamin D toxicity is something called hypercalcemia, which can lead to nausea, to vomiting, to frequent urination, generalized weakness. People can develop bone pain, kidney problems, and even kidney stones from excess use of vitamin D long term. So through sunlight exposure, through UVB exposure, 7-dihydrocholesterol will get converted into D3, which gets taken into the general circulation, and then D2, which is the form we get from diet or supplements, will also get taken into general circulation. And then through the liver, those get combined and then converted into our 25-hydroxy-D, which then eventually gets converted into 1,25-hydroxy-D. And then that goes off to stimulate different cellular activity, either on a non-genomic or a genomic level, which is what we'll get into here shortly. So there's always that possibility, depending on genetic variability, of what may happen with certain people and their vitamin D levels, depending on how much they're either taking, absorbing, or how much is actually being actively produced in the body through these different biochemical reactions. So if you have a polymorphism, for example, in the enzyme CYP2R1, this can compromise overall vitamin D levels. And that particular enzyme is active in the liver, and this prevents the liver from converting D3 into what's called calcidiol, which is technically our 25-hydroxy vitamin D. There's a couple chemical names for it. Now, low levels of CYP2R1 activity have been found in prolonged fasting, in obesity, in type 1 and type 2 diabetes, or even in glucocorticoid use like dexamethasone, for example, which is used in different autoimmune diseases like rheumatoid arthritis. So in these particular circumstances, the amount of vitamin D would need to be taken, oftentimes could be quite high. It could be 5,000, could be 8,000, could be 10,000, could be 15,000, depending on the levels. But again, right, we've got to check to make sure we're getting an accurate assessment. So via sun exposure, diet supplements, we've got different forms of D. The cholecalciferols are D3, the ergocalciferols are D2. Those get converted into our 25-hydroxy-D called calcidiol. And then through a specific enzyme called 1-alpha hydroxylase that is under direction from another gene, the CYP27B1, that converts 25-hydroxy-D into our active calcitriol, which is our 1,25-hydroxy-D. So once all of those steps have happened, we now can engage the vitamin D receptor. So CYP2R1 mutations have been linked to vitamin D deficiency rickets, for example. So this was an article, came out in 2016, that was discussing this. just talking about how CYP2R1 is a hepatic hydroxylase enzyme that helps in the conversion to our 1,25-OHD. Okay, and so the 1-alpha hydroxylase, that actually is an enzyme that's active in the kidneys. So we go from the liver to the kidneys in order to get our active vitamin D. And so if, in certain populations, depending where they are in the world, if there are mutations in this particular enzyme, it can be an increased risk factor for vitamin D deficiency conditions, including rickets. And this was actually specifically looking at risk factor for rickets. So again, another variable of how genetics can play a role in our vitamin D status. So I want to shift gears and talk about something that maybe you don't know about vitamin D. If you've listened to some of my lectures in the past, whether it's through the organic acid test training seminar or some of the webinars I've done before, this is, I think, interesting information. It does have a connection to vitamin D. So let's talk about tryptophan metabolites and looking at serotonin, tryptophan, and quinolinic acid. Now, tryptophan hydroxylase is an enzyme that converts tryptophan to 5-hydroxytryptophan. 5-hydroxytryptophan then gets converted to 5-hydroxytryptamine, which is our serotonin. So there is a connection, obviously, between tryptophan and serotonin through the actions of tryptophan hydroxylase. Quinolinic acid, if you've listened to any of my lectures on the organic acid test, you know that quinolinic acid is linked to a pathway associated with tryptophan. So at the top of this slide, the upper left-hand corner, let me grab a pen, right? We've got tryptophan that gets converted to serotonin through the actions of an enzyme, tryptophan hydroxylase. And we've got quinolinic acid that's sitting down here in this pathway. And one of the things that quinolinic acid does is it helps with the synthesis of NAD, which is nicotinamide adenine dinucleotide. And that's involved in different enzymatic reactions, including the mitochondria. Now, we've known for a long time that there are various inducers of this particular enzyme called indoleamine 2,3 dioxygenase. So viral infections, cortisol, stress, immune stimulation, even a niacin deficiency can increase the activity of indoleamine 2,3 dioxygenase, which pulls tryptophan away from the serotonin pathway into the quinolinic acid pathway. Now, quinolinic acid can, at elevated levels, be a neurotoxin because what it is an NMDA receptor agonist. And these NMDA receptors are in our nervous system, and they're very reactive to quinolinic acid. The other thing they get reactive to is glutamate, for example. And there's different types of neurological conditions, psychiatric disorders, for example, that can be associated with excess levels of quinolinic acid that have a damaging effect long term. Now, this is a complicated slide. I actually use this in one of the lectures I do for the organic acid test training. So if you've not heard this before, don't feel bad. There's a lot of information. I'm not going to go through everything here. But here is our NMDA receptor, okay? And here is quinolinic acid. Quinolinic acid will positively stimulate the NMDA receptor to allow calcium to influx into the nerve cell. The influx of that calcium has a number of other chemical reactions that, for one, for example, could trigger lipase, which starts to break down the cell membrane of the nerve cell. Okay, we get arachidonic acid and we get oxygen radicals that are developed from that. We know that these oxygen radicals can what are called reactive oxygen species. These not only can be damaging within the cell, for example, against the mitochondria, but they can get outside the cell and interact with the glial cells, which are important for regulating synaptic glutamate. Because glutamate, or glutamic acid, also is a positive stimulator of the NMDA receptors. So if we're interfering with glial cells through reactive oxygen species, we get an increase of glutamate, increased stimulation of NMDA, increased influx of calcium, and that too causes a cascade of reactions inside the nerve cell. So one of the things that can happen with a vitamin D deficiency is it allows for more tryptophan to be available for conversion into the quinolinic acid pathway. This was an article, I've used this in many of my lectures too, came out a number of years back. This was specifically looking at autism. But I want you to take some points from this, even if you don't work with autistic individuals, because the biochemistry is the same. We all have what are called VDREs. These are vitamin D response elements that are linked at the genetic level in the nucleus of the cell. We also have different types of tryptophan hydroxylase enzymes. We have tryptophan hydroxylase 2, and we have tryptophan hydroxylase 1. And these have a relationship to the VDRE as well as vitamin D. Okay, so our vitamin D receptor will interact with our 125D, which is our calcitriol. The calcitriol combine, the VDR and the calcitriol combine with another receptor called RXR, which I'll define here shortly. And this complex interacts with these vitamin D response elements that have a relationship at the genetic level, either acting what are as called co-activators or co-repressors of gene activation or gene transcription, which will go off to produce different types of cellular proteins that then have systemic effects, or local effects within the cell or systemic effects in the body. So tryptophan hydroxylase 1 and tryptophan hydroxylase 2. Let's see if I can get rid of this thing here. This control is in my way. Tryptophan hydroxylase 1, let me back up real quick. Okay, tryptophan hydroxylase 1 is found in tissues outside the blood-brain barrier. Tryptophan hydroxylase 2 is found within the brain. Okay, so this is what's happening at the brain level. Over here, tryptophan hydroxylase 1 is what's happening at the peripheral level outside the brain. And technically, serotonin that gets produced outside the central nervous system doesn't really have, at least to any appreciable degree, the level of serotonin inside the central nervous system. Now, there may be some other kind of pathway. We know that the vagus nerve sometimes can be involved, but most of the literature that you read and what's known biochemically, and there may be something out there I don't know about, for the most part, these are two different systems at the blood-brain barrier. Okay, so it's just important to understand that there are two types of tryptophan hydroxylases. So tryptophan hydroxylase 1 is, active outside the brain, particularly in the digestive system. Okay, it plays a central role in platelet aggregation. It's about 95% of serotonin is produced from the gut. And tryptophan hydroxylase 2 is most active in the brain and central nervous system. And essentially, at the blood-brain barrier, there's sort of like a boundary line between peripherally produced serotonin and centrally produced serotonin. I can't say with absolute certainty that that none gets in, but essentially that's the kind of way things are set up. Okay, there's a bit of a boundary. And so what the researchers were discussing in this article is that D obviously has a tremendous influence on these particular enzymes. But because of the way the D reacts with the vitamin D response element and how it responds or reacts at the genetic level and then influences the function of the enzyme, D that acts on the tryptophan hydroxylase, let me back up, D that acts on the tryptophan hydroxylase 2 will cause an increase of tryptophan to serotonin conversion in the central nervous system and a decrease of tryptophan to serotonin production in the digestive system. So if you think about it, what we're talking about here is that there's one of the things that happens when we produce a lot of serotonin in the gut is it can be a trigger for inflammation. So having adequate vitamin D levels is a way to improve serotonin levels in the brain. And so we can think about that clinically. That's important in the autism because serotonin levels in the brain are important for mood, impulse control, long-term planning, behavioral issues, anxiety, even fine gross motor skills. But we know there's a lot of people out there too who, adults, they have nothing to do with autism, have depression issues and low serotonin levels. And just remember that vitamin D does have a relationship on serotonin production in the brain in activation of the tryptophan hydroxylase that is specific to the brain and central nervous system. The other thing I want to talk about is activity at the mitochondrial level. Now, in December 2021, I did a lecture through this service for Great Plains, it's actually on their website, called Mycotoxin-Induced Mitochondrial Dysfunction and Cytotoxicity. So you can watch this, if you didn't see it, you can go back and watch this, because it was a very in-depth lecture. What was interesting, when you just look at mitochondrial dysfunction and associated diseases, the list almost mirrors that for vitamin D deficiency states, because we know that vitamin D deficiency can be linked to diabetes and metabolic syndrome or autoimmune diseases or psychiatric conditions or chronic fatigue or neurodegenerative problems. In fact, I just took a screenshot of the Great Plains lab for the vitamin D blood spot, that section I showed you earlier, right? It's almost a mirror image to some degree. Now, mitochondrial dysfunction is complex. So essentially what's happening is through what's called the loss or reduction in the efficiency of the electron transport chain for the production of ATP is at the heart of mitochondrial problems. And there's three primary molecular mechanisms that induce or reduce mitochondrial function. That would be loss of electrical and what's called transmembrane potential across the inner mitochondrial membrane. We get disruption of the electron transport chain. And then we also just have reduced transport into the mitochondria of different dependent metabolites like NADH, for example. Now, to keep this simple at first, it's important to remember that carbs, fats, and amino acids can get produced into energy. So they basically get sort of funneled down and make ATP. Now, the primary role of amino acids is not to make energy. It's actually to restore our tissue protein levels or the nitrogen pool in the body. But we do take certain amino acids and funnel them into the Krebs cycle, into the electron transport chain for energy production. We get, I should say, the primary effect of carbs is to produce some substrate for ATP production as well as fats. So all of these things can funnel down to make acetyl coenzyme A, which enters the Krebs cycle, which then spills over into the electron transport chain for the production of ATP, that cellular energy currency. And of course, there are a lot of steps along the way that make that happen. So for example, pyruvate, which comes off of glucose, so a molecule of glucose makes 2 molecules of pyruvic acid, which becomes acetyl coenzyme A, enters the Krebs cycle at step 1. And then we move from one step to the next through different enzymatic reactions. And we know that these various reaction steps can also be inhibited either by nutritional problems or various toxins. So heavy metals, for example, can interfere with these steps. When we pull the lens back a little bit on the Krebs cycle, we realize that the Krebs cycle sits in the matrix of the mitochondria. So the mitochondria have an outer membrane and an inner membrane. And there's activity that occurs inside the matrix, inside or along the membrane, the inner membrane, and between the two membranes. And there's a number of connecting points between the Krebs cycle and the electron transport chain. One is through NADH, the other is through succinic acid. The electron transport chain ends in the production of ATP, okay, our main cellular energy chemical or currency that's used throughout the body. Not only inside the cell it's produced, but also we get, that could be cells throughout the body, our nervous system, brain, digestive system, immune system, cardiovascular system. But a lot of the problems with regards to mitochondrial issues occurs at the electron transport chain level. So this is actually happening, or this is embedded into our inner mitochondrial membrane. So we've got, if we look at right here, let's figure this is our inner mitochondrial membrane. And let's just say that here's our outer mitochondrial membrane. So we have an intermembrane space. And that intermembrane space is important because that is the area that accumulates these protons or a hydrogen ion that gets pumped out of the matrix through these different complexes. We've got complex 1, complex 2, complex 3, complex 4. And the pumping of the protons into this intermitochondrial space creates a gradient effect. In fact, what ends up happening is these protons accumulate over top of this ATP synthase. And once that concentration gradient is enough, it's really actually an electrical gradient too, but we get an influx of these protons that moves into this ATP synthase structure. And as it's moving through, we get a rotational effect on these oval-shaped proteins that ultimately bring together adenosine diphosphate and inorganic phosphate to make ATP. So there's a couple connecting points that are linked back to our Krebs cycle, NADH, and then also complex 2 with regards to succinate and our succinate dehydrogenase enzyme. We're going to talk about cytochrome C here shortly, because this also plays an important role in the transfer of electrons along the electron transport chain, because that's what's also happening is we're actually transferring electrons between the different complexes. And that also aids in that pumping mechanism that's involved in transferring out these positive charged protons into that intermembrane space. So there's a lot of stuff happening at the mitochondrial level, which is what I get into in great detail in that lecture I did in December 2021. So we're not only shuttling in different chemicals to be involved in the process of the electron transport chain, we clearly have fuel that also gets converted. So carbs, fats, proteins. Right? We spin things through the Krebs cycle, what's called the tricarboxylic acid cycle. We move things over into the electron transport chain, which is involved in pumping these protons into the inner membrane space. And that ultimately leads to ATP synthase, which allows for these accumulated protons to flow back into the matrix, which stimulates ATP production. So we can have problems that occur at any one of these levels that could affect mitochondrial activity. And that in part is what I was discussing in that previous lecture, how certain mycotoxins can do that. Now, one of the things about the electron transport chain, right, is the transfer or transport of electrons. CoQ10 plays a role in that. Cytochrome C plays a role in that. And when we're talking about these electron transport mechanisms, what's also called an oxidative reduction reaction, we can end up generating a number of free radicals. And one of the interesting things about looking at the mitochondria is that the different complexes are responsible for pumping out of the matrix into this inner membrane space a certain amount of protons. And so actually when we engage the electron transport chain at complex 1, we get the maximally produced ATP based on the amount of protons being pumped out and the amount of ATP that gets produced. Now, there's always a backup system that occurs because our complex 1 could be compromised or our complex 2 could be compromised, which means we can still engage the electron transport chain at the level of complex 3, for example, or even complex 4. But if we start here or start here, we're going to get less ATP produced. Now, again, cytochrome C plays an important role in this, and I'll come back to that here shortly. So just remember that normal activity of the mitochondria is going to generate certain types of free radicals or reactive oxygen species, for example. And that could be a problem if we don't have adequate antioxidant reserves within our cells. And one of those would be glutathione. So glutathione is an important antioxidant to help buffer against oxidative stress. Now, ubiquinone, which is our CoQ10, goes through a chemical reaction as it gets converted to ubiquinol. And there's a radical called a semiquinone that gets produced. And if we, again, don't have good antioxidant reserve, we can end up compromising other aspects of mitochondrial activity by specifically interfering with what's called these iron sulfur clusters. And these iron sulfur clusters are strategically placed in different complexes of our electron transport chain. And they're involved in assisting in the transport of electrons through oxidation reduction reactions. Because remember, as we're moving electrons from one complex to the next, at the same time, we're allowing for positive charged protons to get pumped out into this inner membrane space so that we get this accumulation of these positive charges over top of our ATP synthase. So as these flow back into the matrix of the mitochondria, we produce the ATP. Well, it turns out that these iron sulfur clusters are very sensitive to oxidative stress. Okay, so they can become inactivated by these superoxide free radicals that get developed normally as a byproduct of electron transport chain activity. And if we get too much oxidative stress or superoxide or reactive oxygen species, whatever name you want to give to it, it can compromise overall at various points mitochondrial activity. Now that could be things within the electron transport chain. That could also be areas within the Krebs cycle. Because again, too much oxidative stress will over time deplete glutathione. We need glutathione as part of this equation too, to buffer against these oxygen radicals. And an area in the Krebs cycle that's kind of interesting that takes a hit with disruption of these iron sulfur clusters is the first step, or I should say the first to second step of the Krebs cycle, basically causing an interference in this particular enzyme called aconitase. And if you've ever looked at an organic acid test, sometimes you'll see people who have elevated citric acid. And elevated citric acid can indicate a depletion of glutathione because of oxidative stress that inhibits the ability to convert citrate to this cis-aconitate chemical, that first step essentially of Krebs cycle activity. So there's an interesting article that you can pick up called Vitamin D Receptor is Necessary for Mitochondrial Function and Cell Health. So the vitamin D receptor, okay, we know is involved in binding to our 125D. And then that combination comes together with this, what's called the retinoid X receptor. And that has a relationship in communicating with our vitamin D receptor elements, which then can either increase or repress transcription at the DNA level with regards to various protein development. So we know that one of the main roles of vitamin D, the active form, the 125D, exerts its effect through regulating calcium homeostasis. But obviously it does much more than that. Okay, so the binding to the vitamin D receptor is essential. And then the combination of the vitamin D to the retinoid X receptor is essential as well. And so in general terms, the vitamin D receptor mediates many what are called genomic and non-genomic effects of vitamin D. For this particular discussion, it's important to understand that the vitamin D receptor, through its interaction with vitamin D, helps to protect against excess respiration and production of reactive oxygen species in the mitochondria. So just take what I mentioned prior about the reactive oxygen species effects in the mitochondria, how it can affect the electron transport chain, how it can affect various aspects of the Krebs cycle. So the bottom line is that it can lead to mitochondrial dysfunction. To get even a little bit more technical, the vitamin D receptors will mediate what are called genomic and non-genomic actions. An enzyme, or excuse me, a membrane with this chemical called membrane-associated rapid response steroid will interact at the membrane level with active vitamin D, and that has sort of these non-genomic actions, which I'll describe here shortly, or at least give some examples. But it's the vitamin D receptor that mediates action at the gene level, okay, at the nuclear level, at the genetic level. And so it's interesting when you actually study vitamin D, we realize the interactions and the importance of this chemical are really profound. So another good article for you to get, this one I had talked about up front. This is the article that was giving some of the statistics on the level of vitamin D deficiency in the population, all-cause mortality, 1 billion people, essentially approximately, having a vitamin D deficient state throughout the world. And so what they were talking about in this article is that, you know, around the mid 80s or so, they started to recognize that vitamin D was having more than just effects at the nuclear or the genetic level. there were clearly other responses cellularly that were occurring biochemically, excuse me, that were happening so quickly. There had to be another explanation other than trying to activate things through a VDR receptor at the nuclear level. And it gets very complex. But one of the things that vitamin D is involved in is intracellular signaling molecules. And these are really important because these either can get activated as intracellular receptors that are inside the cell or receptors that are sitting on the cell surface, on the cell membrane. And this is a very complex area of things to study in what they get related to with regards to cell function. So this would be a topic for another lecture for another time. But remember, one of the things we mentioned up front was we all know that vitamin D plays an important role in the immune system. And we've certainly heard a lot about how vitamin D plays an important role with regards to the immune system for viral infections. And so one of the things they describe in this article is that one of the non-genomic effects, meaning it's not triggering things necessarily always at the nuclear level, could be through that membrane-associated reaction, is regulation of the immune system through different cytokines, interferon alpha, tumor necrosis factor alpha. These have a regulating effect on immune responses with regards to viruses, both in immune and non-immune cells. So this particular article goes into a lot of other examples regarding this discussion, but check it out if you're interested in taking a deeper dive into that. But where I wanted to end up with regards to this particular talk is this discussion on cytochrome C. Notice that cytochrome C, so here is our inner mitochondrial membrane. So here is our inner membrane space, and up here would, let's say, would be our outer mitochondrial membrane. So that's that space. And that's where the protons accumulate over top of this ATP synthase, sometimes called complex 5. And that's what makes our ATP. But notice the cytochrome C, which is involved in transferring electrons along the electron transport chain from complex 3 to complex 4, sits on the outer edge of the inner mitochondrial membrane. So cytochrome C is a small heme protein that is loosely bound to that outer surface of the inner membrane of the mitochondria. And it's going to shuttle electrons from complex 3 to complex 4. Okay, and in doing so, that's going to allow a certain amount of protons to get pumped into the inner membrane space through the complex, through complex 4. But the key point is that when the cell, the mitochondria, is under oxidative stress because of too much reactive oxygen species production, or it could be other toxins or something else that's damaging the inner mitochondrial membrane, cytochrome C gets released from its position in that outer segment of the inner mitochondrial membrane. It leaks out, essentially. And that leaking effect of cytochrome C is a trigger, a chemical trigger to the cell to commit suicide. Basically, apoptosis leads to cell death. So there is something quite profound about the cytochrome C and its relationship to the cell membrane that easily gets expressed out of the mitochondria into the cytosolic fluid of the cell, that basically at that point, the mitochondria will say, I'm done. Okay, and if it's significant enough, it also might even lead to just cell death as well. Well, one of the things that the vitamin D receptor does is regulates against reactive oxygen species production, right? We've already established, and I mentioned before, that the vitamin D receptor, through interactions with vitamin D, has a regulatory effect on how much reactive oxygen species gets produced at the cellular level, which indicates that it can have a regulatory effect over cytochrome C and the apoptosis effect of the release of cytochrome C from a damaged cell membrane. So some final comments, and so I want you just to think about these mechanisms and understand, obviously how important vitamin D is. And I really believe, as integrated functional medicine practitioners, that whatever level you are, as far as your training and education, whether that is as a nutritionist or a medical doctor, whatever, right? We've all had various classes that we've gone through, and I think fundamentally it's important to go back and study some of these things from a biological standpoint, from a chemical standpoint, and certainly from a biochemical standpoint, to have a little bit better appreciation for how these reactions occur. And so we know that mitochondrial function and regulation is dependent, at least in part, on the vitamin D receptor and vitamin D. That's been established. What's interesting too is mitochondria act as a factory for inserting iron into different organic molecules. Okay, that, sometimes you just don't think of that way, as the mitochondria acting as a reservoir for different metabolic intermediates. Well, clearly that's the case, certainly if you just look at the steps through the Krebs cycle. So consider vitamin D as an important component involved in mitochondrial function, and maintaining health really means we're also maintaining healthy levels of vitamin D. But it's also important to understand that there is no one-size-fits-all dosage for everybody, okay? What that means is that when somebody asks you, well, how much vitamin D do I take, or how much vitamin D does my child take? Well, we have a range that we can work off, which we talked about earlier. And those can be starting points, whether that's 1,000, 2,000 IUs for a child, or 5,000 for adult, or wherever. But to really be able to hone in for a given individual, we've got to do frequent testing to see what the pattern may be. And so laboratory testing is essential ultimately for monitoring vitamin D levels over time. Okay, so that is my talk here on vitamin D, mitochondrial function, etc. We have a lot of courses through Integrated Medicine Academy, and what we call mastery courses. So we've got a course on autism, we have a course on candida, got 2 courses on organic acid testing, hormones, functional medicine, etc. If you're interested in some of our mastery courses, I'd recommend you go to the website integratedmedicineacademy.com for an overview of the different mastery courses we have. You can also email us if you have any questions at integratedmedicineacademy@gmail.com. We also have a website, which I mentioned up front, beginning of this lecture. This is a membership site for healthcare practitioners called Functional Medicine Clinical Rounds. This is a place that you can schedule one-on-one consults with myself, as well as my partner, Dr. Trenkatella, for lab reviews, for case presentation, case analysis, clinical pearls, etc. We also have some educational material on this site as well, from educational videos to downloadable lab review documents, etc. So more information, you can go to functionalmedicineclinicalrounds.com. And then for those of you needing assistance with certain lab tests, whether that's organic acid testing, other labs from Great Plains, etc., Lab Test Plus is a website that provides access to testing along with a written interpretation of each lab test and the relevant markers. labtestplus.com. You can also email to labtestplus@gmail.com. And then I'm always available for private consultations through my private practice. Here is our website, mysunrisecenter.com. That is our email. If that is something you're interested in doing. Okay, so thanks everybody for your attention. I hope you enjoyed this information. I look forward to the next webinar coming up. I'm Dr. Kurt Wohler. Thanks so much.